Hello, everyone, and welcome to today's webinar titled Functionalizing Genome Editing Techniques on a Broad Range of Cellular Targets. This webinar is part of the Cell Biology virtual event, and I'm Susie Valdez, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on that Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Shondra Miller, an assistant member and Center of Advanced Genome Engineering at St. Jude Children's Research Hospital in the Department of Cell and Molecular Biology. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Dr. Miller, welcome. You may now begin your presentation.
Yeah, thanks so much. I want to thank the organizers for inviting me to talk today. I'm really excited about this virtual event, and I just love that science is still moving forward, even in the middle of what we're going through as a world, as a country together. So as she said, I'm going to be talking to you about functionalizing genome editing events, and I'm going to tell you a little bit about what we do at the CAGE. This is called Center for Advanced Genome Engineering, and we call ourselves the CAGE. So at the CAGE, we basically, oops, I missed the slide. There you go. We have three main missions. First, we provide genome editing technologies for investigator-initiated projects. So we basically help other people at St. Jude do genome editing. We also help we also do independent R&D so that we can stay at the forefront of this emerging and fast-paced field. And then we also support key initiatives such as our gene and cell therapy programs here at St. Jude, particularly in CAR-T and then the hemoglobinopathy areas. We spend about 75% of our time helping others do genome editing, and we do a broad range of editing. In terms of breadth and scope, I would just give you some idea of what we're kind of doing on a day-to-day basis. We provide 6 major services to investigators. I'm going to tell you about those now, and then I'll give you kind of an idea of the numbers of the things that we're doing. I think I'm doing this probably to try to get street cred, but we'll go with it. So first are the services that we provide. We design and validate guide RNAs. We also create custom cell lines, so people can come to us and say, I want to make this point mutation in this cell line, and we'll do it pretty much any cell type and any modification. We also create preclinical animal models, typically mouse and rat. We also do targeted next-generation sequencing for CRISPR-edited samples, and we facilitate high-throughput genetic screens. These are typically pooled guide RNA libraries, knockout, activation, and inhibition. And then we recently started offering edited cell pools, and this is because not every project demands a single-cell-derived clones, which is what we do in the custom-created cell line creation. And we can also get editing rates that are so high that we can actually give them edited cell pools instead of single cell derived clones. So we do a large range of things, but we also do a large breadth of targets. And so just to give you some ideas of numbers, in terms of guide RNAs that we've designed and validated both in RNP and plasmid format, we've really stopped counting because there's just so many, because all our projects really start with that. The next thing, the creating custom cell lines. We've created 495 single-cell-derived custom clonal populations in less than it's been a little over 3 years, I guess, less than 4 years. We have also created 87 animal models. And any given week, we do 4,000 samples per week for targeted next-generation sequencing. We've created 48 custom pooled libraries, and just this year, we've created 44 edited cell pools. So all this to say is that we're doing a lot of genome editing and different species, and then we're also doing it in a wide range of genetic loci. So when we think about genome editing tools, we want something that's going to be super robust and work across a lot of different platforms. So we rely mostly on traditional genome editing. And what I mean by traditional genome editing is that we rely on creating a targeted double-stranded break with CRISPR-Cas9, mostly SP-Cas9. And so you can see that here, we're creating a targeted double-stranded break, and then that double-stranded break gets repaired by one of two major repair pathways in mammalian cells. And that's typically non-homologous enjoining or homology-directed repair. And you probably have seen a slide like this, or you will see one of these today. And I'm not going to go into the details of it, but other than to say that you can use a double-stranded break and the repair by the cell's endogenous repair pathways to create a lot of different editing events. These are targeted mutagenic events. So you can create point mutations, knockouts, translocations, deletions, inversions, all kinds of different things. The two things I really want to bring your attention to are, one, that the repair pathway called non-homologous enjoining is the preferred pathway in mammalian cells. So I've shown this here by this big fat arrow. This is basically just showing you that that happens more often than the homology-directed repair or the HDR events. And then the last thing is that we basically, all of our projects, or most of our projects, start with a double-stranded break. And so we want to make sure that we have guide RNAs that are going to give us efficient double-stranded break creation. So we wanted to basically figure out what assay we wanted to use to figure out when our guide RNAs were actually active. And I'm sure you guys know this, but the guide RNA is the thing that directs the Cas9 nuclease to the specific site in the genomic loci. So we want to make sure that that thing that we're changing out, that guide RNA, is active before we start our project. So the assays that are available for detecting gRNA activity are, there's several of them, and they mostly start with the same upstream pathway. So you basically start out with your guide RNA plus your Cas9. It can be delivered in multiple different formats. Then you're going to transfect that into a pool of cells. These can be, some of those cells will be transfected, some of those cells will not be transfected, some will have breaks, some won't have breaks. Then you're going to harvest genomic DNA, and then do a PCR around the modification site shown here as a red X. And then you're going to want to do one of these downstream applications or these downstream assays. And I'm just showing you some of the most commonly used assays. One is called T7E1 or a cell 1 assay. One is targeted next generation. Then you have TIDE or ICE. And then you also have an assay called IDA. And so a few years back, we wanted to figure out which of these assays was going to work the best for us and look at the pros and the cons of each. And so in work led by Monica Sintmanat with the help of Stan Peters, Colin Florian, and Patrick Connolly, we basically did a survey. And so we published this 2 years ago. It's in Scientific Reports. I'm not going to go into the details of it, but I do want to tell you the take-home message from this paper. Basically, what worked for us, we had several different boxes that we needed to check. And NGS was the one that did those things, checked all those boxes. So the things that we wanted to check were we need to be able to identify active guide RNAs. We need to be able to show indel sizes, how big the indels were. We needed to be able to show sequence identities, what the actual indels were. And then we wanted to be able to accurately portray editing events both in pools of cells and then also in clones. And then lastly, we needed to make sure that we had a very high throughput assay because we're doing a lot of this. We're doing hundreds, tens of thousands probably at this point of different guide RNA validation assays. And so NGS really provided this platform for us. So as with most assays, there are limitations to these different assays. And because all of these assays are PCR-based, remember I told you that the upstream workflow is all similar and it involves doing a PCR step. There are some limitations that are common among all four of these assays. So because it's PCR-based, there is a PCR bias. So smaller amplicons will be preferentially amplified. So large indels will be preferentially amplified. And because it's PCR-based and we have primers that are going to be marking the thing that we're amplifying and sequencing, we can't detect all gross chromosomal aberrations. So large things like if a chromosomal arm got lost or an indel was so large that it deleted a primer binding site, we wouldn't be able to pick those up. Then there's also this limitation of amplicon size. So we're able to basically efficiently amplify off genomic DNA about 1,000 base pairs. And so I would say all of these assays are limited to 1,000 base pairs or less, and you can see the specifics there. The thing that was unique to NGS as far as a limitation was that there's a high barrier to entry. There's kind of, the reagents can be expensive, you need specific equipment, and there's expertise required in not only setting up the library, but also analyzing the data that comes out. So that can really, scare people off of doing NGS. And so just to give you an idea of what a genome editing workflow looks like for us and for most people, and followed by NGS, I wanted to give you a workflow of this. So first, we'll basically introduce the guide RNAs. It can be into cells or into embryos to make those preclinical animal models, mouse or rat. Then you allow the guide RNAs to do their thing, and you culture the cells. You can culture them either as single cell clones, or you can plate them as pools of cells. Or you can even put, if you do the embryo injections, you can put them back into a mom and let the mom do the culturing while she's pregnant. Next, you're going to harvest genomic DNA. You're going to do a PCR around the region of interest, and then you're going to add indexes so that you can pull all the samples together, sequence with NGS, and then analyze your NGS data. And we do this with something called CRIS.py, which I'll tell you about in a few slides. So going into more detail about how we do our NGS library setup, we have dual indexes, and we use a two-step PCR setup. So what this looks like is you would have your region of interest. This is where your guide RNA is cutting in between these two primer sites. You would have a forward primer and a reverse primer that are specific to the locus of interest. And then you would have these orange overhangs that we call these partial Illumina adapters on the 5' end. You would amplify in the first step, you would amplify the gene-specific region, and then you would put on the partial Illumina adapters. And then in the second step, you would use those partial Illumina adapters to do a nested PCR and put on your indexes. And in our lab, we have 44 forward primers, and then we have 384 reverse primers that are common that we use across all different projects. And the forward primer has an index of 6 base pair, and the reverse primer has a 10 base pair index. So what this means is that we can do a whole bunch of PCRs. We can take them from wells of plates. We can figure out what index goes to which well, and then we can demultiplex when we pull all those samples together. And so you get results, an amplicon that looks like this. You do traditional NGS. We use Illumina's MySeq platform for most of our applications. And we are able to deconvolute those indexes and figure out where the editing event is occurring in which well. So we get this result that looks like this. You get a whole bunch of FASTQ files, and this is pretty scary for most people. It was scary for us the first time we saw it, and we're like, what are we going to do with all this data? There's lots of data. It takes up a lot of space, but how do we actually get stuff that we want out of it? Inside those FASTQ files, you can see the efficiency of the editing, you can see the size of the indels, and you can actually see the sequencing identity of what's going on with your genome edit. And so I like to make this joke because my mom was a huge X-Files fan. The truth is out there. I should really probably say the truth is in there. Inside those FASTQ files, you can really find the information that you want to see with the limitations that I expressed earlier. So in a Herculean effort, I guess, Patrick Connolly in the lab, he basically is a biologist, trained biologist, had no formal experience in coding. He basically learned Python using Codecademy online, a free online coding course, just so that we could analyze our NGS data efficiently. And he came up with this pipeline called CRIS.py. Cute name. We like the name because Python programs, this is written in Python, end with .py. And then we also could make this cool little logo on the bottom left that has a Python in it with the S. So we like the name, I guess you can tell. So we have this paper published just last year. So if you want more details, you can go there. But I'm also going to walk you through this paper in a little bit more detail so you can understand some of the things I'm going to tell you based on the analysis that we have. So to be completely upfront, there are quite a few NGS analysis programs out there. And to be completely honest, most of them are written more eloquently than ours, because again, ours was written by a biologist for biologists. So it's very simple and it's highly functional. And so you can totally use one of these other analysis pipelines, but I'm going to show you some of the advantages that CRIS.py has and the reason why we still use it in our lab. I guess we developed this about 7 years, so we've been using this for about 7 years now. The things that we needed our NGS pipeline to do were the following. So we needed to make sure that we could run it locally. We didn't want to rely on somebody's server to actually be able to run our NGS analysis. If it goes down or they take it down for maintenance, we would be stuck. So we needed to be able to run it locally. We need to be able to run it on lots of different samples. We're doing thousands of samples every week, so we couldn't just do 1 or 2 at a time. We need to be able to do a lot at one time. We also need to be able to look at more than one donor search at a time, so more than one edit happening in individual clones and across multiple clones. And then importantly, we needed a master summary file so that we could look through a lot of editing events and be able to figure out what we actually want in that, in the needle in the haystack kind of scenario. How do we find the needle quickly? And so to go over how CRIS.py works, again, written by a biologist for biologists, so it's super simple. We basically take the sequence, the FASTQ files, and we treat it like text. And so we start out with a reference sequence, which is basically just the amplicon that you sequenced around the modification site. And then we ask the program, we give the program a start sequence, or seq start, which is basically a 10 to 14 base pair sequence on the 5' end of your reference sequence or your amplicon. Then we also give the program a 10 to 12, 10 to 14 base pair sequence that we call the sequence end at the 3' end of your amplicon. And then we ask the program to measure the distance from the start seq to the end seq, just literally as characters. How long is the seq start to the seq end? So let's say in this example, our sequence from seq start to seq end was 250 base pairs. Then we would tell the program to look for anything that had the start seq and the seq end, and then to measure the distance compared to the reference. The reference length is 250 base pairs, and our sequence that matched the start seq and end seq is 249 base pairs. It would report it back as a -1. If it's 250 base pairs, it would report it back as a 0, and so on. And so that will report out the indels. And you can see here just showing the indel length. So 0 again would be the wild type length, and then you can see if they're missing or adding base pairs, it would be reported back. And because we're not only doing knockouts where we're looking for indels, we're also looking for knock-in modifications or modifications made by homology-directed repair. And so what we can do is we can give the program a test sequence. So what this means is we say, okay, any sequences that have the start sequence and have the end sequence, how often do they have this exact match to a test sequence? So you can think about this as saying Control F in your FASTQ files of how often do I find the thing that I'm actually looking for, if you think about like a word processor document. So those are the things, that's how it works. We basically look at length, how long is the sequence that we're looking at in the reads compared to the reference, and then 2, how often do we find the thing that we're looking for. So as far as CRIS.py input, in this example, I'm going to show you where we were looking for the activity of these 2 different guide RNAs where we're putting in guide 10 or guide 14. In terms of input, what you need to do is basically just put 6 things in. And this is how the program is available on GitHub as command line. And that scares people. It scares me. I don't even know really what command line means except for this part. But I'm a programmer and I feel comfortable using this. What this means is you're going to go through and these 6 things on the left, the text that's written or highlighted in red, you're going to change the text in between those apostrophes. So the things you're going to be changing are, number 1 is just giving it a name. So if I wanted to call it locus 1, I could call it locus 1. I could call it VHL if I wanted to give it the gene name. So whatever you want to call it. Then you're going to put in your reference sequence, which again is just the amplicon, the sequence between those 2 primers. And then you're going to give it your seq start, that small sequence at the 5' end, your seq end, which is that small sequence at the 3' end, and then you're going to leave 5 alone because it's going to be you're going to look in all of your FASTQ files, and then you can give it a list of as many test sequences as you want. So in this case, we basically gave it 2 test sequences, so guide 10, so the full 23 base pairs, including the PAM sequence of guide 10, and the full 23 base pairs of guide sequence for guide 14. That's it. 6 things, really 5 things that you have to input, and one of those is just the name. I also want to mention when we were doing when we had reviews for this paper, one of the reviews is they didn't like that it was modifying the command line. That's not like how programmers typically do things. And so Jake Steele in the lab, he's a new member of the lab, he actually made something called Pretty Crispy, which has a graphical user interface or a GUI, so that you can do this more robustly. This is limited, that you can't do it as many samples as what we're talking about, but it is out there for folks if they want to use it. And that's also available on GitHub. All right, so we talked about the input, we talked about how it works, what does the output look like? So this is a master summary file. Remember I told you that was one of the things that we really wanted to be able to do, is to find that needle in the haystack very efficiently. And so I'm going to walk you through. We have 2 outputs. One is the master summary file, which is just a CSV file or an Excel document, basically, that is sortable and searchable. And on the left, you have here the FASTQ file name. That's the well in which the reads actually occur. So we use those index that we did in our 2-step PCR to basically go back so we know, okay, this sample was in plate 1, B07. So if the hit that we are looking for is in plate 1, B07, we can go directly to that and expand that clone. The next column is labeled sample, and this is user inputted. So this is actually blank besides the column header, and you can put in whatever sample name you want just to be descriptive for your own purposes. Then the next column is total, which is the total number of reads that you have for each of the samples. So the first sample, our negative control, had 980 reads. And then we're going to go into our test sequences. So this is that list of sequences. Remember in the previous slide, I had G10 and I had the 23 base pairs of the guide 10 target site. So we're looking for how often does that guide 10 site occur with 0 base pairs of mismatch, where it basically is a perfect match. And what you can see here is that in our negative control, we have almost all the reads match back to guide 10. It's not going to be 100% because if there was a sequencing error in any of those base pairs, it would not be considered an exact match. So it's never 100%, but that's why we always include a negative control so we can see what the sequencing error is for any of our different test sequences or our links. But then you can see with guide 10, you're losing that guide 10 site. So that is a good indication that we're getting the modification that we're looking for. The next column is called total indels, and this is a quick and easy way of checking to see how much activity you have, because it's going to report back the percentage of reads that are not wild type links. So again, in our example, I told you that the wild type link was 250. So anything that's not 250 will be reported back in this column. So I can quickly and easily see that guide 10 is the most active guide RNA and out of the two, guide 10 and guide 14. And then after the total indel column, you can actually see the top indels and their frequencies. So on the right, you can see it says number one indel, number one dash reads. So the number one indel is a 0. So that means in all of the samples, actually 0 is the most common indel. So no indels created. This link is the wild type links. And then you can see as you go on, you can see in sample G10, number 1, you can see that indel number 2 is a +1, and it occurred at 15.7% of the time. And then at the very end of our master summary or CSV file, we have some QC checks, which you can look into the paper. I'm happy to discuss in the Q&A section, but I'm going to kind of ignore for right now. The second file that we output with CRIS.py is a text file. And if you think back to the master summary file, you may have been like, okay, this is good. It's got the summary of what's going on, but I don't see any sequences. The sequences are actually outputted in the text file. And in the text file, we're going to give you the top exact matches and the number of frequencies of those reads, the number of times those reads occur in each of the wells. So in this case, I'm showing you the negative control first, that plate 1B07 negative control just transfected with GFP. And I've highlighted for the presentation the guide 10 and the guide 14 with their respective colors, and the arrow indicates where the double-stranded break would occur. And so what you can see here is that I can immediately look through, and if you look on the right-hand side, you can see that the length of these amplicons are all the same. And you can see that the top one has 644 reads, and that exactly matched that. And then after that, you go down to only 11, then 10, then 9 reads. And what this is sequencing error. So anytime a base pair is not exactly matching, it's going to be binned separately. And so you can easily go through, and I've highlighted in red those sequencing errors. So no cutting, no editing in the negative control, which is what we would expect. Then if we look at our experimental samples where we put in guide 10 or guide 14, I can also immediately check by looking at the length or the size of the amplicons. You can see on the right-hand side, it looks kind of like, I call it the river, it kind of looks like a river, because the amplicon sizes are no longer the same length. And you can look to see that what the sizes are. So a +1, a -2, -15, and so on. One thing I do want to point out is that with CRIS.py, because we're looking at either lengths or exact matches, in the link section, it's going to bin things that are the similar length together. So in this example with guide 14, there's actually 2 different -45. So you see -45 at the top circled at 23%. If you go down to the guide 14 text file on the bottom, you can see there's actually 2 different two different -45 indels. So 2 distinct indels are binned together in the master summary, but you can parse those out in the text file. So it is important to look at both the master summary file and then the text files to know exactly what's going on in each of your wells. CRIS.py also allows you to look at multiple HDR events, and you can analyze them concurrently. And this is important, and we do this actually more than you would think. In this example, we wanted to make a heterozygous modification in which we had what we call a clean het, where one allele does not have an indel in it, and one allele has a modification. So in this example, I'm showing this thing that says mod C to T, and we basically wanted to make the modification that's shown in red. But we also wanted to make sure that the other allele we didn't want homozygous clones, we only wanted to be heterozygous to recapitulate what happens in the patient population. And so what we did is we put in a blocking oligo, or what we call block ssODN. And what this is we made a silent modification highlighted in red, the C, where we basically changed a base pair in the wobble position of the amino acid. So we changed the codon, but not the amino acid that's being expressed. And so what this will do is it will trap that second allele so that we don't get an indel event on it, or once we get an editing event, it doesn't have a subsequent cut and an indel event. So in this case, what we're going to do is we're going to put a guide RNA in, the Cas9 in, and then we're going to put in these 2 different blocking modifications. So 2 different ssODNs, one the block mod and one the blocking oligo. And so looking at our CRIS.py analysis, we can quickly look at our test sequences where we put in about 10 base pairs on either side of the block mod ssODN and then also of the blocking ssODN. And we can tell that the block mod showed up at 13.4%, and then the block only showed up at 10.4%. So we know that we're getting the editing that we want. We can also look at the indels. The total indels looks like 52.8. And then on the right-hand side, you can see the top indels. And looking at our text file, it's easy to see in our negative control, one major read, some subsequent sequencing error. Then you can look in our edited sample. You can see the wild type read is the top read. And then after that, you can see our block mod and block sequences, and then the indels showing up after that. Oftentimes, we aren't creating pools. We're actually wanting to go through single cell clones, and you can actually get more bang for your buck. You can get several different clones or several different types of genetically modified cells from one round of screening. So in this case, we were able to use our master summary file to quickly and easily find the clones that we were looking for and some bonus clones. So I've highlighted just 3 different outcomes. So there's Roman numeral 1, 2, and 3. In green, you can see wild type plus block mod that's basically the genotype of the clone. And we know this because G6 is basically the wild type sequence, and it's about 50% G6 and about 50% of that blocking modification ssODN. So those equal about 50-50 to 100%. The modification 2 is the block mod block. And again, in yellow, you can see that the block mod ssODN test sequence and the block ssODN sequence are about 50-50. And then for number 3, down at the bottom in orange, you can see that we also were able to get knockout clones where we have all the alleles are out of frame. And we also got some other things where we got wild type clones that we could use as controls. We also got some that were heterozygously knocked out with 1 wild type allele and 1 knockout clone. So you could use these all as controls. And if you know what you have quickly and efficiently, you know which ones to expand and freeze down. All right, so one of the things, I mentioned that there was all these other NGS analysis pipelines, but one of the things where I think CRIS.py really shines is in this master summary file, and really it's the only pipeline out there that I know of that actually has a master summary file. I wanted to compare this, the output of CRIS.py to CRISPR-2 because it's one of the most widely used analysis tools, and it's beautiful. It gives you these beautiful graphs, beautiful pie charts. I love it in a lot of different ways, but we still use CRIS.py because of this master summary file. Just because one of the reasons is the output. So for CRISPR-2, for every sample, for every amplicon, you get basically 123 files per amplicon per sample. And so if you had a 384 well plate, which is what we typically would screen for a knockout or a knock-in modification, you would get 47,000 files. So this is a lot of files. It would be very hard to sift through to figure out which clone has the modifications that you want. In comparison, CRIS.py basically gives you 2 files, and that's regardless of the number of amplicons and regardless of the sample number. And so if you had a 384 well plate, you'd have 2 files. If you had 10,000 wells, you would have 2 files. And so it makes it a lot easier. And our master summary file is a CSV file, which is basically a, sorry, phone's ringing, basically an Excel document that is searchable and sortable. And if you have any more questions about this, it is published, and we do have this CRIS.py video tutorial. If you can find the link in the paper. All right, so I've gone and walked through kind of what we're doing at the cage, this broad range of editing and lots of different, both in animals and also in cell lines and screens. We're doing all this different stuff, and we're using NGS and CRIS.py as our pipeline. So I thought I'd give you some of the tricks and tips that we've learned over the last 8 years or so, and hopefully this will help you with your experiments. So one of the things that we've learned is that NGS can really help you determine the allelic frequency. And so what I mean by this is that a lot of times sometimes people come to me and I'll say, okay, you want to knock out this gene, how many copies are there? And they say, oh, it's human, it's got 2 copies. And I say, oh, well, is it, a cancer-derived cell line, or do you know that it's only got 2 copies? And people are sometimes surprised, but it shouldn't be, that most cell lines that we use in lab are polyploidy. And they can actually be very polyploidy, especially for some specific chromosomes. So in this case, we were able to use NGS to figure out that in N2As, there's actually 7 copies of this gene. And we did this by looking at the frequency of each of those indels. So on the bottom, I've colored the different indels, and you can see that there was a -14 that occurred at 18%, -5 that was at 26, a +1 at 27, and a -4 at 27, 28. And we were having a hard time wrapping our minds, like, how many alleles is this? Is this truly a clone that we want to keep? And if you take 100 divided by 7, it's 14. And so what we found is that basically, we had 1 -14, 2 -5s, 2 +1s, and 2 -4s. And you can see that aligned here. There's 2 -4, 2 distinct -4s, and 2 distinct +1s. So you can use that ratio to figure out how many copies you have without even doing debanding karyotyping in your clones. And this is fairly easy to do, especially for triploid and tetraploid clones. It gets a little harder when it's over 4 copies. Another thing that I think is a little bit surprising to most people is that within a population and culture, it's quite heterogeneous in terms of allelic number. So, you know, sometimes you'll see debanding, or you'll see something printed, the karyotype, and says, okay, the cell line is diploid or the cell line is triploid. When you actually go through and you start looking at hundreds of clones you see in that population, you actually have diploid, triploid, and tetraploid clones if it's said that it's triploid. So here's an example where I've just highlighted in green the triploid clones, and I did this because if you look at the indel ratio, so for example, 18B09, the second one down, the first green one, you can see that there's a -11 at 63%, which is roughly 66. So basically 2 alleles had a -11, and 1 allele had a -1, which is represented around 33%. So all the green ones are triploid. The red one is tetraploid because you have basically 50% -4, +1 at 23%, and a -2 at 23%. So basically 50, 25, 25. And then you also have diploids that are at 50, 50. And then you have some clones, actually quite a few clones that are homozygous where you don't really know how many alleles because all the modifications are the same. So you can determine the heterogeneity within a cell population. And this helps us out. We try to help the end user by picking clones that represent the population the best. So in this case, we had mostly triploid. So we would have picked triploid clones, and we would have picked clones, 2 clones. We try to give them 2 clones. We would pick clones that have the same zygosity if at all possible. That limits some of the variability downstream. Also, sometimes genes you can't knock them out. They're essential to the viability, or a cell line is dependent on a specific gene. And so in this case, we were not able to get a clone that was completely knocked out, but we were able to find dose response clones. So I'm going to show you, have a star. Basically, we couldn't knock out all 3 alleles, but we were able to knock out 1. So you can see this -8 and indel 1. And you have 1 wild type, which is the 0. And then you also have a -6, which I'm calling, that's why I have the asterisk, because it's wild type, because it's just missing 2 amino acids. So it probably expresses like wild type, and it's in-frame mutation. We were also able to find a clone that had 50-50 expression. So on the bottom, you can see there's a -17 and a 0 at 50-50 ratio. So that's going to have 50% of the protein expression potentially knocked out. And then we also, oops, wrong way. We also have a clone that has just 1 allele remaining, 1 wild type allele. So you can actually look at the dose response of having less and less protein in the cells if there's no compensation. And that's something that you can easily do with our CRIS.py program at NGS. Also, we use NGS to genotype pups to know which pups to breed, which pups have the modification that you want, and which pups have them at a high ratio, that modification at a high ratio. So in this example, we're just trying to create a simple knockout animal. I've highlighted in green the indels that are in frame, and then I've highlighted in red the indels that are out of frame. And what you can see is maybe we would definitely choose not to do A1 because A1 has 2 in frames, and that won't create our knockout modification. But pup A4 has 2 different red indels, so we might use that one because we could breed it and get 2 different lines out of it if you want to have 2 independently derived lines to compare. All right, another thing that we've started doing with our NGS analysis is looking for dependencies, and this is really, we do this for 2 different reasons. One is because we're trying to knock out, sometimes people give us a gene and we think, oh, this might be essential in the cell line. It looks like it is. We're getting mostly in-frame mutations. And when we screen the clones, and so we can actually go back and do this dependency or fitness assay. And I'll explain it in just a second. And then the second reason why we use this is to follow up on hits from knockout screens, where we say, okay, if we knock out this gene and we apply the stress, it causes the cells to either increase in proliferation or decrease. And so we can use that to follow up and validate hits from screens, essentiality screens. So what does this look like? This fitness assay looks as follows. So we basically would have a cell line. So in this case, NOM6, wild type cells. And red is going to be the wild type links. So we're saying that 97% of the amplicons when we did that PCR were wild type links. And then there's that black bar is the out of frame indels. And then we'll take 2 different guide RNAs. And this, I'm showing you the first set of guide experiments. So this is guide 3, and we take a sample at day 3, day 5, and day 7. And what you can see is the gray is the in-frame mutations. The black is the out of frame mutations, and then the red is the wild type link amplicons. So what you can see is that between day 3 and day 10, after RNP transfection of your guide plus Cas9, you don't really see a change in the allelic ratios or the indel ratios. Then we also take a second guide. So this case guide 10. So we do 2 different guides to make sure that it's not just guide dependent. And then we can also see at day 3, day 5, day 10, you don't really see a change in the in-frame, out-of-frame mutations. So this gene is not dependent. This cell line is not dependent on this gene. And it would be, you would be able to knock it out. We also, in this case, here's a dependent one where we take the same cell line. We take a guide RNA called guide 4 to a new gene, gene Y. And you can see that over time, you lose that black bar, which is the out-of-frame indels. And then we do it with the second, do the same thing. So you can see that you're losing it over 50% reduction in the out-of-frame indels. And so this is easy to see that this looks like it's basically validating a hit from a screen or showing you that it's unlikely that you're going to be able to get a knockout cell line. Now, it is we call it a fitness assay because it could just say that when you knock out that gene, it causes a severe growth disadvantage, but it's not essential necessarily, but it causes a severe growth disadvantage. So I mentioned earlier that we use Cas9, SP-Cas9 predominantly, and we do this because it works really well across a broad range of cell types, cell lines. It's got not a very restrictive PAM with just 2 GGs next to each other required. And it works most of the time. So I would say 90–95% of the guides that we use give us high activity. But there are times where we use other Cas9 orthologs. So I guess our second go-to Cas ortholog is Cas12a. And there are some differences between these. But the reasons why we like Cas12a is because it also works very robustly. It's commercially available. You can actually get the protein. So we like to deliver our reagents as RNP if possible. And then also it's got a T-rich PAM, which is nice because it complements the G-rich PAM of the Cas9. So we can cover more sequence. Something else that I want to point out that I don't know if a lot of people think about is Cas12a has a longer spacer region. So Cas9 only has a 20 base pair spacer, 1920 base pair spacer, whereas Cas12a has a longer, so typically around 25 base pairs. So what this does is it allows you to, if you're looking at sequences that are highly homologous to other sites in the genome, you could maybe use a Cas12a to give you a more unique guide RNA or more unique cut. And so here's an example of, and I put in NGS as platform agnostic because you can use NGS for base editing, prime editing, different Cas9 orthologs or Cas orthologs. In this case, I'm going to show you an example where you use Cas12a. So in this example, this gene is highly homologous, or this region is highly homologous to several other genes in the genome. So when we look for Cas9 NGG PAM, we couldn't find one that was unique. We like to have at least 3 base pairs of mismatch to any other site in the genome, and we couldn't find anything that was unique. But when we went to Cas12a, which has the longer spacer region, we were able to find a guide RNA shown here. The PAM sequence is over here, so it's on the 5' end of the guide RNA shown here, 3' end of the sequence shown, sorry. And then it's TTTV. And so what we want to do in this experiment is we wanted to insert this high-bit tag directly in front of the stop codon. And what we were able to do is use Cas12a to find it. We found a guide RNA that was unique and had 2 base pairs of mismatch, so it was much more unique than the Cas9 alternative guide. And so we used this and we introduced the Cas12a protein, the guide RNA, and then also this ssODN to insert this HIBIT tag. And what you can see here is this blocking modification that we also introduced. Again, we introduced it into the PAM mutation, or into the PAM site, so the first T of the TTTV. And then we also put it in the wobble position, so this lysine is not changed. And so our CRIS.py analysis looks like this. So we have some negative controls, GFP only, then each of the oligos by themselves. So we did this antisense and the sense. When I say antisense, I just mean the bottom strand and the sensing the top strand here. And then we also put in the nuclease plus those ssODNs. So I can quickly go to my total indel column, and I can look and say, okay, the first 3 are negative controls really have no cutting. That's just sequencing error. And then whenever we put the Cas12a, you can see that you've got a lot of cutting, over 95% cutting, 97 and 98 in each of the wells. So they cut fairly equivalently between the 2 different samples. And you can also see that the indels, indel number 1, indel number 2, indel number 3 are the same across the 2 samples. So you get consistent cutting across the 2 different samples in the cell type. And then the other thing we're going to look for is that specific modification that we wanted. So we put those as our test sequences. And so one of our test sequences is guide 1. So we're looking to see how often does that guide RNA site match exactly. And our controls, you can see it's very high, around 90%. Or higher. And then in our experimental, we can see that guide 1 goes away almost completely down to 0.6 and 1.8%. And this is because we had really high editing, 97, 98% editing indels, and then we also had our hibit tag going in with that blocking modification. The second test sequence is going to be just that blocking modification by itself, and then the third one is the hibit tag, the sequence shown in yellow, which is 33 base pairs. So what we can also see is that the antisense oligo works better than the sense oligo. So whenever we use a strand that was not the targeting strand as the oligo donor, it works better. So if we look at the hibit tag column, you can see that there was 1.8% integration of the hibit tag when you use the antisense strand, but only 0.1% whenever you use the sense strand. And so, of course, we went forward and we single cell sorted the antisense sample, and we were able to find homozygously tagged clones shown here, 7B9 and also 8D2. And I just wanted to show you how this looks different from the pool versus the clones. Now, when we look at our block only or our hibit tag, you can see high rates of those. And you can also see that the indels are 100% because we've knocked in this 33 base pair hibit tag. All right, so another thing that we often do is we do create large deletions. So I'm going to give you an example where we created a large deletion, 150 kb deletion in the cell line. So we wanted to delete all the regions shown here in gray. And so we put in 2 guide RNAs, Cas9 protein, and then we screen our pool to see if the deletion happened by doing this PCR where we have a gene, we call it Gen F and Gen R, so they're out primers. So these primers are about 150 kb apart when they're not deleted, when the section in between is not deleted, and they're only 350 base pairs apart whenever they are deleted. And so you're only going to get amplicons whenever you have the deletion of a product. And so we actually run these by NGS as well. And what we do, our little trick that you can use, is we use something called a bridging oligo again. And what this looks like is we basically just take 40 to 60 base pairs on either side of each of the guide RNAs. And then we make a single-stranded oligo donor, so that basically our hope is that basically when you get the double-stranded break, single-stranded end is exposed, and it complementary base pairs to that end, and it kind of bridges it to the other double-stranded break. And we wanted to test this if it's actually helping or not. It was an idea, but we didn't know it was actually helping. So what we do is we put in an ECAR1 site, so this GAATTC on the bottom. We put that in to see, is it happening from just the rejoining, the 2 breaks rejoining, so not anything to do with the ssODN or the bridging ssODN, or is a guide RNA, or is the ECAR1 site being integrated into that cut site, which shows us that the ssODN, the bridging ssODN was used. And so here's just 2 examples. This is the first example where we basically use guide 42 and guide 3. Also, I'll say that there's only 1 sample coming up on our NGS data because, again, you only get an amplicon if the deletion has occurred. So all of our controls are going to be no reads. So they won't show up on our NGS analysis. But in our experimental, where we put all the business in, all the stuff that we needed to make the break and rejoin it in, we actually saw no perfect deletions. We did see 78% of our rejoining events, that deletion events, were actually incorporating the ECAR1 site. And we can also see this in our top indel because 6 base pairs is the indel size, the most prevalent indel size. When you look at when you align the sequences from the text file, you can see that our top read is that GAATTC being integrated, that 6 base pair and the ECAR1 site. And then you can see we got a lot of indels that didn't rely on the bridging ssODN, but they are not perfect rejoining. In this case, it would be fine to use that because we were just trying to get an approximate deletion occurring. And that's why we were okay with the ECAR1 site. If you wanted a precise rejoining event, you could use an ssODN, a bridging ssODN, and not put that ECAR1 site in. We were just doing it to see how often the cell was using either just rejoining or using the ssODN to fix those breaks. Perfect rejoining does occur, and I think the smaller the deletion, the more likely you don't need a bridging oligo. This is an example where we're doing about 100 kb. So when I say small, I'm talking like 2 kb or less. The bridging oligo becomes less important. This is about 100 kb deletion that we're doing here, but we did see some perfect rejoinings. In this case, it was 4.6%. But again, the bridging oligo helped a lot. Most of the deletion events actually included that ECAR1 site, over 50%. All right, so then I want to talk a little bit about large insertions. In this case, we're using single-stranded DNA, so something that's just a single-stranded piece of DNA that we typically have made. And we typically use 200 base pairs of homology arms. So on the 5' and the 3' site, just 200 base pairs on either side of what you want to insert. In this case, we're basically inserting this T2A CRI upstream of the stop codon. And so you can see that shown in the bottom in the red. And what we would do is what we call junction PCRs, where we have a primer that's outside of the homology arm, gene-specific, gene F, and then we also have a junc R. We call these junction, where it's basically a primer landing inside of the insert. Then you would do each of these PCRs, and then you could submit for NGS, because these amplicons are about 400 base pairs. With NGS, we're really limited to about 500 base pairs in sequencing with Illumina. So what you might do if you weren't using NGS is you could easily just run this PCR out on a gel, and you'd see something like this. So here's our 5' junction, here's our 3' junction. You can see that we have 13, I think 13, maybe 15 pups, 16, I don't know, something around there, 15-ish pups. And you can see that most of the pups are positive. If they're positive for one, they're positive for both. You can see that this pup here missed the 5' junction, so you wouldn't use that. But other than that, you can't really distinguish between. With NGS, we basically run this PCR, and then we throw it onto our NGS run for the week, and then we get the sequences back. So I'm going to show you what the sequence looks back for the 3' junction. For the 3' junction, we actually have a test sequence that goes across the junction, shown here in red. And what I can immediately see is pups 1 and 2 do not have an exact match to that 3' junction. So something is off in these pups where they don't have a precise rejoining. Pup 4, however, has a high rate of that exact match. You can also see pup 2 has a 98.8% indel. So it has something that's not even the right length. It's not an exact match or the right length, but it does have the junction. So when we look at this in more detail, we can see that in the very bottom, which is pretty tiny now that I see it on my laptop, you can see that there are some mistakes here. We see this more often with, we see this really only when we use ssODNs or ssDNA, and it's been reported in the literature. I think we talked about it with Steve Ecker's group, where when you're using ssODN, something about the repair process is less efficient, where you have some mistakes at the cut site. We also see, if you look down at the other pups, some of the lower pups, you have point mutations down here, shown in red on the right-hand side. Those, I don't think those have to do with the repair. I think those are more likely a synthesis error, something to do with the long ssDNA construction that had a mistake in it, and our pup just happened to use that as a repair template. But the good news is that we did have some pups, these ones starred in green, that were precise, and they had the exact modification with no other errors or no mistakes in the repair. Again, another thing that is good about NGS, but a limitation is that we could not sequence through the whole thing. We could only sequence through the business end. And since I talked to you about these synthesis errors, it's really important to sequence through the whole insert before going forward to breeding this animal. All right, just a few more slides. I wanted to talk to you about this on-target activity limitation of PCR bias. So I mentioned this. PCR bias is seen easily whenever we're dealing with animals, because we know that in the F1 generation, mom can only give one allele and dad can only give one allele, so we know that it should be 50. So this example, we did embryo injections, found a pup that had a -33 base pair deletion. That was our F0. Then he was actually mosaic, so he had more than just 2 alleles, but he can only pass on one whenever you made him to I can't remember if it was girl or boy, made him to a wild type black 6. And so we know that the allelic ratio in the F1 generation should be 50-50 of whatever we have. But when you look at the NGS data, you can see pup 31, the very top one, had a -33 indel at 61%, and then wild type from the black 6 parent at 36%. So that larger deletion made the amplicon about 10% smaller. So the PCR, the polymerase, preferentially amplifies the smaller indel. So be aware of that whenever you're doing any PCR-based assay, whether it's NGS, or some IDA or TIDE or ICE, shown there. And then I talked about amplicon size and how you're limited to really about 1,000 base pairs. You can get kind of creative with this and put more things into your NGS run. One way we do this is whenever we use double-stranded DNA, which is what we typically use for creating cell lines. We use junction PCRs like I described earlier, but we have, in the double-stranded DNA, we typically use about 800 base pair homology arms, which makes this 5' junction and the 3' junction amplicon about 900 base pairs in length, which is too big for NGS. So what we do is we'll do actually 3 PCRs. So we do the first PCR, which we call the long junction. Then we take that PCR and we do another PCR, which we call the short junction. And then we do our indexing PCR. So in this case, our short junctions, you should only give an amplicon if you have the target integration. And our short junctions will be 400 base pairs, which we can sequence. So in this case, you'll only get reads from the wells that have the junction, where you actually have the long junction. And so you can do this, since our output is in Excel, you can either create something in Python, which is what we did, or you can do something in Excel where you compare how many pups had the 5' positive, how many pups had the 3' positive or cell lines clones, and you can actually figure out which ones have both the 5' and 3' junctions to know which ones to narrow it down fairly quickly. And so in this example, 62% of the clones that had the 5' correct junction also had the 3' correct junction. And then I also mentioned earlier that there's a high barrier to entry for NGS. We hope that CRIS.py has helped with the, you should not be intimidated by it. CRIS.py, again, was written by a guy who just learned how to do Python coding for this particular application, so it's super simple. And then the cost is the other thing that's a barrier. So what we've done to make NGS cost effective and actually cheaper than Sanger sequencing in our case are several things. And I just wanted to share some tips. One is the more samples, the better. So we sequence anything we can sequence, we throw into our weekly sample. So we sequence gRNA plasmids, we sequence pools, we sequence clones, we do junctions, deletions, translocations. We even do STR profiling to identify cell identity. So you can put anything you can think to put in there, you should put in there, because the more the better. You can also pool samples. So we basically pool the samples and we only do one run a week. So we basically take all the samples that we can get all over St. Jude, and we run one run a week so that we can get the most bang for our buck. You can also consider trying a spike-in program. So if you don't need a lot of reads, but several other labs are interested in getting some reads, just the reagents cost from Illumina for the MiSeq run is about $400, and you get 1 million reads. The MiSeq, sorry, that was the nano run. The full run, you get 15 million reads, and it's only $1,200 for the reagents. So you get 15 times the number of reads, and it's only 3 times the cost. So if you can get the more people that you can get doing sequencing, or the more samples that you have, the cheaper it becomes. We use a two-step PCR because it's generally more cost-effective versus like a TruSeq where you have to use specific primers. And then we try to make our setup as quick and easy as possible. So we do robotics when possible on the PCR. We use crude lysis for the genomic DNA, and we use a multi-drop to just fast dispense the reagents. And so my last 2 slides are really just going to kind of sum up this notion that NGS can actually be cheaper than Sanger sequencing. So hopefully it's not going to be a barrier for you to enter into this spot. So I mentioned that a MiSeq full run is $1,200 for the reagents. I get that there's a machine and a service contract also involved, but just the reagents are $1,200. You get about 15 million reads typically. If you do 1,000x coverage, which is overkill for most of the things that we are doing in our lab, you could do 15,000 samples, and that will basically be 8 cents a sample. So it's much cheaper than Sanger sequencing, even if you add in the cost of the service agreement and the labor involved in hiring somebody to help run this. Let's say you don't need 10 million reads or 15 million reads. You can do a nano kit, which is about $400. You get about 1 million reads. If you want 1,000x coverage, you can do about 1,000 samples. And this is only 40 cents a sample. So still, again, cheaper than Sanger sequencing, and you get a lot of information. So in conclusion, I hope I've shown you basically 3 main things. 1, NGS is an excellent on-target assay for CRISPR activity. It's also good for off-target activity, but we use it really to figure out, do we have the modifications that we want? Also, CRIS.py is this Python-based program that is freely available on GitHub, and it allows even novice users to interrogate their data completely. Like, you can really dig down and figure out, what was the cell doing? What did the cell do in my cell? It's not a black box that's really hard to understand. It's very simple. And again, I've trained lots of scientists on how to use this and interpret it, and it's very simple. And then finally, NGS is a high throughput method for quickly, cost effectively, and quantitatively measuring your editing outcomes. And then I'm going to leave you with thanking the people who did all the work. So I basically started the center, the GEIC at Washington University. Monica Sitmanat was the one that led the survey of the validation assays with the help of Patrick Connolly, Sam Peters, and Colin Florian. I also mentioned Patrick several times because he was the one that developed the CRIS.py Python program. And then really, we're very much a team. So everybody in both organizations was involved in the data that I presented today. And then also to thank the funding. And I'll leave you with this picture of the folks in my lab now who are working diligently every day. And then I'll take any questions that we may have.
And thank you, Dr. Miller, for that informative presentation. We will now start the Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. Dr. Miller, we already have some great questions coming in. Our first audience member says, thank you for your helpful presentation. Based on your experience for knocking out a bacterial virulence gene, that contains both the conserved and the hypervariable regions, which part is better for targeting?
This is a question that I probably cannot answer because we haven't done that. I guess I assume that they're talking, well, I'm not exactly sure what they're specifically asking, but typically whenever you're knocking out a gene, I mean, you could definitely delete the whole thing, like the two-guide approach, if you're worried about it. But I would say most of the time, if you get an indel in there, it'll mess it up. It'll cause it not to be expressed. But I would say if you're really worried about it, just do a deletion where you delete the whole thing out. If it's going to be small, most genes are 1,200 base pairs or less if you're looking at the cDNA. So I would say deletions of 1,200 base pairs are fairly efficient, so that would be no problem at all.
Wonderful, thank you. We have time for one more question. Is CRIS.py available for download? And if so, how can I download the program?
Yeah, absolutely. So I would encourage you to go to the paper that we published. So Patrick Connolly's the first author. CRIS.py. I think if you search for CRIS.py and St. Jude, you'll find it. You may find something about fried chicken. I'm not sure. But yes, you can absolutely download it. There's a link to get the GitHub site that also has a link to the pretty CRIS.py where you can use the GUI, the user interface. Absolutely, we wanted to make it available to everybody. And if you have questions, we're happy to help. But yeah, it's definitely available, and there's that tutorial too that can help you in your first goes at it.
Dr. Miller, thank you again for your time today and for your important research. And before we go, I also want to thank our audience members for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information provided at the time of registration. And this webcast can be viewed on demand for 6 months until March of 2021. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, stay healthy. Bye-bye.